Welcome to this new video. In this video, we will see post-docking analysis. So I will cover complex preparation, non-bonding interaction, besides hydrogen bond surface, charge, hydrophobic, ionizability, 2D and 3D interaction. So these tasks will be carried out by PyMol and Discovery Studio Visualizer. So for post-docking analysis, first we need to complete our molecular docking. After the docking, we will obtain or get some files named as PDBQT file. This PDBQT file for protein and these are for ligand. How you will obtain this PDBQT file, you can see in another video where I have shown how to carry out molecular docking and how to find this PDBQT file. So I have accumulated this PDBQT file in a folder. So this is for protein and these are for ligands. I have taken two ligands, so they are showing two ligand PDBQT file. So first task is preparation of complex. For complex preparation, we need the PyMol software. So you can open the PyMol software from your start button or as this PDBQT file. As this PDBQT file open by default by the PyMol software, just double click on this protein PDBQT file. It will open in the PyMol software. So after open the protein chain, then we need the ligand PDBQT file. So you can add this ligand PDBQT file from the file section, then open and then ligand one. So ligand is added. You can also add your ligand from the file directly. Just click on this ligand and drag it into the PyMol software. So it will add in with your protein chain. So as we have already added our ligand, now further we need to save this protein and ligand combination. So go to the file section and export molecule, save. We will save this complex in this folder, give a name CX complex 1, and you should add it in PDB format, then save it. So our complex is prepared. You can also prepare your complex by ligand 4 with your protein. As we have as we have make our complex, now further we should see our non-bonding interaction. So for this analysis, we need the Discovery Studio Visualizer software, as the PDB format by default open with the Discovery Studio Visualizer. Just double click on this PDB file. So you can see that this is the protein chain and this is the ligand. So this ligand is binded in this pocket. Now we can see the protein and ligand interaction. For that, go to the display receptor ligand interaction section and click on the ligand interaction. So ligand interaction with the amino acid residue of the protein is showing. So these are the non-bonding interactions with the protein. So you can also see the non-bonding interaction type. So for that you need we can change our visualization from the script option visualization. You can add publication quality visualization. Okay, then we can also change the display style from that. As usually this format is widely accepted, so I have added this display style. Further, Uh, you can also change some settings for clear visualization and for realization. So we don't know what is the amino acid residue in this section. So you can also add the name of the amino acid. For that, right click, go to the labels, add. So we will add the name of protein, amino acid residue. Right on, click on amino acid attribute. We will give three letter and ID number and font Times New Roman. Times New Roman. Font size will be 18. Color is black. Then press OK. So you can see that the amino acid residue name is also showing here. So you can see that the benzene ring is create a non-bond interaction with the leucine 352 and also with the alanine 527, and the fluorine atom here create an non-bonding interaction with the methionine and here with the valine. So you can see that in our schedule non-bonding interaction we have seen the non-bonding interaction, but we don't know what is the bond non-bonding interaction, bonding distance, and also the type. So for that, we need to go to the below section, click on this arrow sign, go to the non-bond, click on this. Now you can see that the non-bonding interaction type, distance, category are showing here. So first, the color green color bond. Here you can see that this is the green color bond with the oxygen atom with serine residue. This bond is so the bond category for this green colored non-bond. You can see that the bond category is hydrogen bond and the type is conventional hydrogen bond and the distance is 2.50, and the valine and fluorine interaction. This fluorine is the fluorine of ligand, and the protein amino acid residue is valine 523, and this bond is hydrogen bond, halogen bond. So types is carbon hydrogen bond, and the distance is 3.52, and the methionine 522 and fluorine bond is halogen bond and type also halogen, and there are also some hydrophobic and pi alkyl group. So for your manuscript you can and add this residue involving in the non-bonding interaction and the distance and category types. So if I want to show how to add this value into your manuscript, you can see that these are the residue in contact. These are the distance of this ligand with this residue, and these are the type and category. So I will copy these all values from this dataset. So select these values and Control C, copy. Then paste it in these values in a Excel sheet. Then you can see that these three column are not required for us. So I will delete these three column as these are the residue and bond distance category type. So I will delete these three column. So select these three column, then delete this. So now these are the residue in contact and these are the distance. So I will copy this residue and paste it in this table. As you know that for manuscript there is required a standard representation or table. So I have prepared a table. Headings are ligand, docking score, residue in contact, interaction type, and distance. So, and this is the ligand 1, ligand 2. These ligand are binded with the protein with a specific protein, and I have already added the docking score of this ligand with this protein, and these are the additional values associated with this docking score. So at this moment I am working for ligand 1. So I will paste this residue in residue in contacts column. So you can see that these are given an additional information with the residue. At this moment I need the others character except the serine 530 for manuscript or for a standard paper. We need just the um, residue name and the residue number. So I will keep the serine 530 and others information I will delete it. You can also keep the ligand section, ligand fluorine atom interacting with the methionine 522. But I at this moment I don't need this values except this residue in contact. So I have cleared the other information except the residue and their number. So copy this residue and paste here. Then the distance should be in this column, and these are the hydrogen bond category, but here I am not seeing the category section. I'll create an category. So bond category are these values, paste these values, and these are the bond type. Now you can see that ligand 2 will be moved in this row, and this ligand should be in the merge cell, and also the docking score in the merge cell. So this is the representation of the interaction type with the protein residue, amino acid residue. So then you can copy this table into your manuscript or in a your manuscript, or you can also paste it in a Microsoft PowerPoint for your further designing or a better representation. So you can add your interaction type of the ligand with the protein into your manuscript. Now we have seen the non-bonding interaction. We will see the 2D and 3D interaction hydrogen bond surface. So these values are not required at this moment. You can see that this is the 3D structure of the ligand, and these are the interactions. So this interaction is 3D interaction type, and you can save this interaction into your manuscript by moving this figure and getting a best configuration or position of this interaction type. So I think this representation or configuration is not this position. I think this representation is the best for describing the all interaction with the amino acid residue. As you can see that this oxygen atom is a non-bonding interaction with the serine 530, this benzene ring with the alanine and leucine, and these fluorines with the valine and methane. So you can take a screenshot for representing into your manuscript. If you have an already installed screenshot app, then you can use these tools. So I have already installed this screenshot app. Then copy this and paste it into your Microsoft PowerPoint for further designing. Okay, so this is the 3D figure. You can also see the 2D diagram. So by click on this show 2D diagram, you can see the 2D interaction type. So this is an excellent figure for adding in the manuscript. So you can also see the there is given the interaction type, and this is serine and oxygen interaction is conventional hydrogen bond. This is an halogen bond. So you can also save this figure by right click, then copy and paste it into the Microsoft PowerPoint. You can also take a screenshot by your screenshot apps. Okay, so we have seen the 2D and 3D interaction. At this moment, we will see hydrogen bond surface charge, hydrophobic, ionizability. So for that, you can see that I will minimize this. So you can see that in this tool, display receptor surface, here you can see that this is aromatic, this is hydrogen bond, and this is charge, hydrophobic, ionizability, SAS. So this surface you can add into your manuscript depending on your analysis type. So I will see the hydrogen bond. This is the hydrogen bond surface. So you can take a screenshot by your screenshot app, and this is the parameter. These position are more hydrogen bond donor, and these are the acceptor position. So by screenshot app, you can take a screenshot and represent these values. Sorry, this figure in the PowerPoint. So when you take a screenshot, you should be concerned that the mm, uh, the other part except the structure covering the screenshot should be cropped. So crop the other part. Okay. So this is a best figure, and you can add this 3D figure, this hydrogen bond interaction, and also the ionizability or hydrophobic. These three combination figure you can use in the in your manuscript by giving a name, the ligand name, or you can also add the 2D interaction type. Okay, so we have seen charge, hydrophobic, and ionizability. So these values using depends on your analysis type. So that's it for today. Thank you.